changed. So when you think about evolution, you typically think of a process that takes millions of years. And my research group are really interested in understanding the fundamentals of evolution. And so to do this, we see evolution happening in real time in the lab using bacteria.
Bacteria have a really short generation time, so you can watch them evolve in front of your eyes. You tinker with something, you run the tape again, see how they evolve. And that's fantastic because you get to basically do experimental evolution, where you're watching these new phenotypes arise in front of your eyes that have massive consequences for the bacteria.
So model systems are something that's really important to the way that our research is designed. We use two broad model systems. One of them is involved in understanding gene network rewiring. So essentially what we do there is we break an essential gene, we put it under selection for the recovery of that gene, and we see how the gene network reassembles to rescue that lost function.
So the model system I use takes Pseudomonas fluorescens. We disable its ability to produce flagella, the little tails that let them swim. We stick them under a selection pressure for motility, where they have to swim or starve. My day-to-day life involves taking a colony of these immobile bacteria, placing them onto a plate of agar where they're able to swim, but they've got barely any food. Within about 3 days, we'll see these bacteria re-evolve motility, re-evolve the ability to construct their flagellum. They'll start spreading out from the middle of the plate. They'll have taken another gene from the same family, plugged this into their network, and used this to rewire their whole network. And my research really looks at why they use some genes to do this and not others.
So using this model system to understand gene rewiring, we've been able to discover that there exists certain rules that determine which genes it can borrow to replace that lost function. And something else that we've been looking at is understanding how bacterial defense systems, which allow them to form an immune response against viral infection, how these defense systems are maintained and evolve within a novel host.
So my research focuses on how phage, the viruses that attack bacteria, influence the evolution of those bacteria that they infect, and in particular how they influence the evolution of the defense systems the bacteria have to fight against the phage. So one of the cool quantitative analyses that we're able to do is at the population level, we watch the bacteria grow over time. So you can watch, if there's no phage, the bacteria does a classic growth curve, and if you add the phage, they can maybe try to go up, and then you just see them flatten out and just not grow at all until right at the end, you'll see them start to take off in growth again as the absorbance increases in the media, and that's them evolving resistance to the phage in front of your eyes over a 24-hour time series. So why I think my research is important is, as we're facing down the antibiotic resistance crisis, there's this turning notion to try and use phages as a new method of bacterial control. So if we can understand how bacteria gain resistance to phage and what the interplay is between the two, we can hopefully design better treatments that are basically more like evolutionarily robust.
Well, the future ambitions of the lab group are to build in greater ecological complexity, and this will hopefully allow us to understand these evolutionary processes within the context of more complex microbiomes. I found Bath to be a really supportive environment to build my research career. There's excellent expertise across microbiology, molecular biology, evolutionary biology, and it really encourages these interdisciplinary conversations between the groups that I found have really helped my research group thrive. It also attracts brilliant talent, so building my research group from scratch here has been a real pleasure, and I've been really inspired by a lot of the research that's gone on. So I've just found it to be a supportive and encouraging environment to build my career.